We are in topic 2 focusing on topic of fruit anatomy.
The standard consumer definition of a fruit relies almost entirely on sensory experience. If a plant product has aromatic flavors and naturally develops a high sugar content, we categorize it as a fruit and serve it for dessert. Conversely, the culinary classification of a vegetable is dictated by how it is prepared. If an edible plant is heavily cooked and served as part of a savory meal, it gets grouped into the vegetable aisle. Relying on a culinary binary creates massive blind spots in food science. Categorizing commodities by taste profile means grouping items with completely different cellular structures, moisture contents, and decay rates, making it impossible to standardize industrial processing and storage methods. To build a rigorous framework, we have to look to botany. The strict botanical definition states that a fruit is exclusively the edible product of a plant, consisting of a seed and its developed envelope, derived from a ripened ovary. Vegetables, on the other hand, do not belong to any specific botanical grouping. The term acts as a catch-all for a wide variety of vegetative plant structures, including roots, stems, and leaves. This diagram illustrates exactly how scattered the vegetable classification is. We group a swollen taproot like a carrot in the same category as an underground bulb like an onion, or a stem tuber like a potato. Because of the heavy bias toward flavor in culinary definitions, countless true botanical fruits end up falsely classified as vegetables simply because they lack sweetness and feature prominently in savory dishes. Basing post-harvest technology on how a chef categorizes ingredients leads to rapid spoilage and inefficient supply chains. The only reliable way to manage and preserve plant commodities is to categorize them through a standardized morphological taxonomy. Regardless of its external appearance, thickness, or flavor, every true fruit begins its life cycle in the exact same location, within the protective confines of a flower's ovary. The developmental sequence is triggered mechanically. Once pollination occurs, the ovules inside the flower transform into seeds, which sends chemical signals instructing the surrounding tissue to expand. As it matures, the primary structure of all fruits resolves into two distinct parts: the seeds themselves and the pericarp, the botanical term for the fruit wall. The massive diversity of fruits across the globe, from a delicate raspberry to a heavy pumpkin, is simply the result of variations in the structure, thickness, and moisture content of this pericarp. By treating the pericarp as a modular architectural system, we can categorize every single fruit based precisely on how its distinct layers differentiate during maturation. To classify any fruit biologically, we must map the structural differentiation of its three-layered pericarp. This cross-section of a peach serves as our primary architectural model, demonstrating the relationship between the internal seed and the fruit wall. The outermost layer is the exocarp, or epicarp, the plant's protective glandular skin mediating interaction with the external environment. Directly beneath that is the mesocarp. In most fleshy fruits, this develops into the thick, succulent, highly edible portion that attracts animals for seed dispersal. Finally, the innermost layer is the endocarp, forming the final physical boundary layer that securely encloses the seed itself. By tracking how these three specific layers thicken, harden, or merge into one another, we can logically derive every major category of fleshy fruit. The first step in establishing a high-level taxonomy requires determining exactly how many ovaries were involved in the fruit's initial formation. We define simple fruits as those derived from one single ovary. As this chart indicates, this category includes a vast array of forms, from avocados to tomatoes. Aggregate fruits, by contrast, are those derived from multiple independent ovaries contained within a single flower. And multiple fruits are the composite products of many completely separate flowers packed tightly together. Pineapples, blackberries, and raspberries all develop through this clustered reproductive strategy. From here, we will focus our analysis exclusively on simple fleshy fruits. This is the group where the entire pericarp, or a portion of it, remains succulent, juicy, and indehiscent at full maturity. Look closely at these tomatoes. While a chef calls them a vegetable, botanically, they serve as the definitive example of the berry subclass. The defining biological trait of a berry is that its mesocarp and endocarp remain entirely undifferentiated. They merge to form a unified pulp in which multiple seeds are completely embedded. The second major subclass is the drupe, commonly referred to as a stone fruit, which provides a direct structural contrast to the berry. In a drupe, the endocarp heavily differentiates from the rest of the flesh, hardening into a dense, stony layer that strictly encloses a single protected seed. There are specialized variations of this design. In a coconut, the mesocarp develops into a dry, fibrous husk rather than juicy tissue. The white, edible portion we consume is actually the endosperm feeding the seed. This structural deviation dictates entirely different processing pipelines. Industrial facilities require specialized pitting blades to process drupes, equipment that would completely destroy a unified pulp berry. The morphological matrix includes highly specialized pericarp modifications, such as the Hesperidium, the characteristic fruit type of the citrus genus. The Hesperidium features an outer glandular skin for its epicarp, while its mesocarp develops into a spongy, fibrous layer known as the pith. The actual edible portion consists of hundreds of juicy hair outgrowths that swell and fill the compartments defined by a membranous endocarp. Another major structural variant is the pepo, a classification that encompasses large commodities like cucumbers, squash, and melons. A pepo is defined strictly as a large, fleshy fruit developing from an inferior ovary with parietal placentation, meaning the seeds attach to the outer walls of a deeply pulpy, unilocular interior. Because Hesperidia and pepos feature complex, multi-chambered interiors and dense, fibrous boundaries, agricultural engineers must design precise humidity and moisture control strategies to prevent rapid internal rot during post-harvest storage. The final structural anomaly in the fleshy fruit group is the pome, a subclass that includes apples and pears. A pome is technically classified as a pseudocarp, or false fruit. In this mechanism, the flower's receptacle develops simultaneously with the ovary. This non-ovary tissue swells outward, becoming the massive, fleshy, edible portion of the apple. The ovary itself is pushed inward, forming the tough, papery core that contains the seeds. The core is the actual true fruit. Understanding this structural inversion allows food scientists to manage the competing decay rates of the external receptacle tissue versus the internal true fruit, maximizing viability in commercial transport. The morphological structure of the pericarp directly correlates with how a fruit behaves physiologically once it is detached from the parent plant. This table outlines the respiration rate for a range of commodities. Respiration is the primary physiological indicator of deterioration. The higher the rate of carbon dioxide production, the lower the item's keeping quality. Items with a low respiration profile, such as nuts and citrus fruits, inherently retain their quality longer in storage than high respiration items like aggregate berries and avocados. To accurately predict shelf life, this structural and respiratory data is filtered through one final classification layer, determining whether a fruit is climacteric or non-climacteric. This chart plots physiological activity over time. The red curves track climacteric fruits like bananas and tomatoes. They exhibit a massive post-harvest surge in ethylene gas production and respiration, allowing them to fully ripen after being separated from the parent plant. By contrast, the flat blue lines represent non-climacteric fruits like grapes and lemons. They produce minimal ethylene and must remain attached to the parent plant to reach full physiological maturity. By discarding arbitrary culinary labels and mastering the botanical matrix, from pericarp differentiation to respiration profiles, agricultural scientists can reverse engineer the exact environmental conditions required to secure the global food chain.
Thank you. Disclaimer. Artificial intelligence, AI, was used in the development of selected instructional materials in this lesson. The use of AI is intended to enhance clarity, visualization, and learning engagement, while all academic content has been reviewed and guided by the instructor.